Hello, everyone. We are honored to be here. We have really loved meeting all of you over these last days, and now we'd like for you to meet our project, PHOENICS. Imagine the year 2050. Many diseases of today will be treatable, but one will only increase as we live longer: cancer. Already, it's the second leading cause of death worldwide. For decades, cancer treatment has relied on chemotherapy and radiation, essentially poisoning the body to eliminate the tumor. Recently, cell therapies have emerged as a powerful alternative. Among them is CAR T-cell therapy, where one harvests a patient's own immune cells, re-engineers them to better target the tumor, and then re-injects them. This has shown remarkable success in blood cancers, proving that with synthetic biology, we can reprogram cells to fight cancer. But CAR T-cells have several downsides. The need for patient-specific cells makes them difficult to scale, driving costs to over $200,000. Even in blood cancers, immune exhaustion can hinder CAR T efficacy and result in relapses. And finally, in solid tumors, which make up 90% of cancer cases, immunosuppressive environments and high diversity limit CAR T-cell success. So, we asked ourselves, what if we could create a new kind of cell therapy, one that's off the shelf, safer, and applicable to all cancers, including solid tumors? We call it PHOENICS, a platform for a new generation of cell-based therapies. To make this possible, we had to think differently. Instead of re-engineering existing immune cells, we built a bottom-up synthetic immune cell. So, we went to the drawing board to design our system. We needed tumor-specific sensing, fast and reversible processing, and flexible responding, all integrated into an orthogonal chassis cell that is not affected by immune exhaustion and can be mass-produced for off-the-shelf use. In May, we took this idea to Heidelberg's largest innovation competition. Winning it not only showed us public interest, but the potential our project holds in revolutionizing tumor therapy. Here is how we turned our idea into science. First, our cells need to know where they are. To achieve this, we use two receptors, one that turns the system on in the tumor, and another that turns it off in healthy tissue. After consulting experts in synthetic receptor engineering, we adapted two receptor architectures and turned their switch-like behavior into potent tumor sensors. But sensing tumors isn't enough. Our cells need to compute these opposing signals and decide: activate or stay silent. For that, we built a dynamic phosphorylation circuit. This is how it works. Our receptors are coupled to either a kinase or a phosphatase. When the stimulatory receptor is activated, it recruits a synthetic substrate, which is then phosphorylated. In healthy tissue, the inhibitory receptor is active and dephosphorylates the same substrate. Only when phosphorylation outweighs dephosphorylation, our cells activate. And phosphorylation is fast, responding within minutes, as well as reversible, meaning our cells are constantly adapting to their environment.
But building cells isn't just assembling parts. We need to know what ratios of receptors work best, at what ligand concentrations does the cell activate, and how strong is the phosphorylation level. To answer that, we built our digital twin, SPARC. SPARC models how our receptors, enzymes, and substrates interact to predict how PHOENICS cells will behave, allowing us to fine-tune our circuits before we ever enter the lab. This massively accelerates development, enabling us to adapt PHOENICS to any tumor ligand profile. Now, with this in hand, the only thing left was a therapeutic output. For this, we added a phosphobinder protein, which only attaches to our substrate when it's phosphorylated. By coupling a split transcriptional activator to both proteins, we were able to translate phosphorylation into expression. Following expert advice, we even integrated a more rapid secretion platform based on the split TEV protease. When phosphorylation activates this protease, a pre-synthesized protein is released from the ER. We even improved the flexibility of the system by secreting interleukin-12, a potent cytokine. Finally, we put the full circuit to the test to see whether sensing, processing, and responding can work as one integrated unit. First, we transfected the entire PHOENICS circuit with concentrations optimized by SPARC into hex cells. Then we induced our results with two opposing ligands, TNF-alpha, a soluble protein, as the stimulatory ligand, and rapalog, a small molecule, as the inhibitory ligand. These inputs were computed by our receptors. The signal was then computed by our phosphorylation circuit, leading to expression of a luciferase. After 24 hours, we quantified this using luminescence. And here's what we saw. We see a twofold increase in luminescence when we added TNF-alpha alone, but when we also added the rapalog, the signal dropped back to uninduced levels, completely reverting system activation. Now this is exactly the kind of smart, adaptable logic we need for a synthetic immune cell. But here's one more thing. To truly target any cancer, we needed a pipeline to generate, validate, and deploy new tumor sensors. To demonstrate this, we targeted GDF15, a protein upregulated in pancreatic cancer. First, using Bindcraft, a deep learning model, we generated entirely new protein binders. From there, we ranked them using molecular dynamic simulations by predicting their binding affinities in silico. This pre-screening helped us identify our strongest binders, saving us time, cost, and resources. Next, we moved to wet lab validation, where we selected our top candidate using in vitro testing and then integrated it directly onto our GPCR receptor. And here's what's remarkable. We actually saw activation of our full phosphorylation circuit in response to GDF15. This pipeline is what makes PHOENICS scalable. We can now target any cancer, whether it be pancreatic, cervical, glioblastoma, and integrate them into our off-the-shelf cells within days.
Now, let's take a step back and look what we've built. PHOENICS is not just one therapy. Every component, from the receptors to the logic circuit to the effector modules, can be swapped, tuned, and combined. SPARC, our digital twin, predicts how these parts will interact before we ever enter the wet lab. We offer this part collection to the iGEM community, inviting future teams to build on our vision of a programmable synthetic immune cell. Our circuit architecture can be implemented into many chassis cells. Take, for example, mesenchymal stem cells. These cells are non-immunogenic, meaning they don't have to be re-engineered for each patient individually. Down the line, we even imagine our circuit in a fully synthetic chassis. But proof of principle is only the first step. We are missing a clear path from the bench to the bedside. This is why we sat down with leading experts across research, regulation, and patient advocacy to turn our questions into conversations. To share what we learned, we hosted a panel discussion on cell and gene therapies. Through active dialogue with our high-profile panelists, we and other students learned about the obstacles in translating research. The feedback was clear. The next generation of scientists wants to shape the future of translation. However, through the event, we realized that many young researchers, including ourselves, did not know where to start. Through numerous conversations, we found that our region actually offers a strong, helpful ecosystem. But this ecosystem is limited. It is fragmented in some regards, and most often students are not aware that these programs even exist. So we built the Heidelberg Translation Roadmap, a clear student-facing map which gets a team from where to even start to a concrete first meeting. It includes upcoming events, insights, workshops to get a team started. This map has not only supported our journey, but most importantly, it will enable future teams to start their journey. And we're keeping it alive. We're constantly updating it with new upcoming events and growing a local community around translational research. So, here's our vision for the future. Let's say a patient is diagnosed with pancreatic cancer. Then, a biopsy is taken and secretome analysis maps a precise tumor profile fingerprint. From this profile, we can then select suitable up and down-regulated targets. Using SPARC, we can then design and optimize the best-fitting circuit architecture and implement it into mesenchymal stem cells. After quality control, these cells are then ready to be infused into the patient. But here's the best part. These cells now join a growing library of PHOENICS cells, ready to be redeployed to the next patient with a similar secretome profile. So, step by step, we engineer a future where synthetic immune cells outsmart cancer.
In other words,
The circuit is built.
Let's switch it on.